so much for the invitation. It's a pleasure to be here. And I always love to bring it back to, and now for something completely different, because that's one of my favorite comedy acts. But what I want to talk today about is using viruses as carrier molecules, because we all know how wonderful viruses are for inducing appropriate immunity. And as you've seen that this morning and this afternoon. I've worked with my scientist, Jyoti Melachosula, and Dr. Webby at St. Jude Children's Research Hospital, who's done the challenge studies for these experiments. So it's not advancing. So what we've heard about repeatedly today is that the best immune-inducing molecules are the ones that mimic the molecules that carry the infectious agent. So the best vaccines come from replicating or live attenuated viruses that all have the capability of inducing pattern recognition through either toll receptor or antigen cross-linking on B cells. What we also know is that the nucleic acid is an incredibly powerful way to also induce immunity. But what we also know about these viruses is that it's difficult to make safe live vaccines. And there have been a lot of efforts to use nucleic acid to carry the antigen into cells. And what I'm going to talk about today are subunit protein vaccines, which are probably the worst possible antigenic stimulus that you could imagine, because they're too small. They often are not do not carry the ability to cause antigen cross-linking, and they don't have toll receptor stimulation capability because they're typically devoid of nucleic acids. So the middle intersection here is non-replicating viral vectors have an ideal balance between safety, because they can't usually pick up and re-replicate and make a pathogen, and they also have these both nucleic acid and antigen cross-linking capabilities. But yeah, so proteins don't have either of these. And how do we take what we know about proteins, the things that nature has identified as important for specific immunity, and confer upon them these characteristics that make them good antigens? So obviously, virus-based vaccines, they have excellent innate immunogenicity. They're replication competent, packaging defective. We've heard about those today. They're attenuated to reduce pathogenicity. They're inactivated, and influenza vaccines that we get seasonally are a great example of that. But especially for influenza, production requirements can be extremely onerous. One of the biggest issues with influenza is production in eggs. Often the yield is low, and part of that often has to do with the characteristics of the proteins that you're trying to drive expression of in the attenuated virus. And the recombination events in some cases limit the ability to use a specific virus strain. Lentiviruses are especially problematic, but the risk for this outweighs the benefits of using those as hosts for vaccine development. But subunit vaccines, they're typically easier to produce than a virus. Some production systems can make up to 5 to 10% protein weight in an expression system. They're high yield, they're low cost, they're easy to purify, they're safe because there's no risk for recombination or a virus infectivity, and they're often the agent that is defined by nature as the target for neutralization of a pathogen. So HA is a great example of a molecule on influenza that was identified because that's what patients that survive influenza make antibody responses against. So you can specify and limit the immunity to the exact thing that's required for neutralizing the viral infection in vivo. Yeah, and they're terrible antigens. They're typically cannot be given without formulation. They require adjuvants, which also become problematic because of cold storage or because of toxicity or because cost. So what I've been working on for the last 2 decades is using TMV as a tobacco mosaic virus, which is a plant virus, as a carrier molecule for vaccine development. And one of the interesting things about TMV is that it's also been modified genetically to make antibodies, and the ZMAP antibodies that were just discussed in the previous talk are actually produced in plants using a modified TMV viral expression system. So not only can TMV molecules be used as vaccines themselves, but they can also be used to drive expression in plants. So TMV is a rod-shaped virus. It has a very defined diameter and length. It's probably one of the most oldest characterized viruses because it was the first one that was named. It was the first one that was purified. It actually was the first virus that was called a virus. It was the first one for which X-ray crystallography was available, and it also has a repetitive structure that allowed, ironically, for the definition of the alpha helix of DNA. So it's a very historically rich virus, and there's a lot known about it. It's also extremely easy to grow because you can make it in plants. It expresses at 5 to 10% total weight of plants. So if I grow a kilogram of plant tissue that's infected with TMV, I can get 5 to 10 grams, which at 10 to the 13 particles per milligram is a lot of material, and it's very easy to produce. So mass amounts are no problem to produce in plants. So we can also genetically modify the coat protein because it's a single molecule, and it's a very small molecule, and the genetic basis for TMV modification is actually very simple. Unfortunately, it doesn't replicate in mammalian cells, but that's also a benefit because there's no risk for TMV to replicate in a mammalian host. We've actually played around with the RNA core to see if we can get it to replicate temperature shift mutants, but at this point, we don't really have a TMV that will replicate in mammalian cells. You all probably ate some TMV today in your salad. Every human being on the planet has been exposed to TMV orally. You all have antibodies against TMV. There was a study done recently that surveyed 2,000 people, half smokers, half not, and the theory was that the smokers would have antibodies to TMV, but actually everybody does because it's in the food chain. And so the assumption is that because you've been eating it and you've been exposed to it, it's not particularly harmful, it's not going to hurt you. So we also did a lot of studies early where we fused peptides genetically to the surface of TMV by coat fusions. And we showed that we could get both antibodies and cytotoxic T lymphocytes that would protect immunized mice from tumor challenge in several different models, as well as L2 peptides from papillomavirus that would protect from papillomavirus tumors. So what we've been working on more recently is how does TMV actually provide vaccine activity? And the first assumption was this is a rod-shaped virus. It's not like any other viruses that your immune system would see. Why is it a good immunogen? So our first studies were to look at mouse and human primary bone marrow-derived dendritic cells or blood-derived human dendritic cells, and then we pulsed them with an Alexifor-labeled TMV and used them as we trypsinized to make sure that we weren't just looking at sticking, and we used flow cytometry to look at uptake. So this is mouse dendritic cells. After 24 hours, you can see you get a large population of cells, and at 96 hours, almost 90% of cells in the reaction have taken up fluorescence labeled TMV. And one of the things that you can't see is that the mean fluorescence intensity also increases dramatically. So dendritic cells aren't just taking up 1 or 2 particles, they're taking up so much so that they almost can't be measured by flow cytometry. They're too bright. They smash up against the right side of the axis over here. We also saw the same thing in human dendritic cells, where you can see that you have an increased level of uptake. So it's not just mouse, it's human. I think it's a characteristic that if it's the right size, dendritic cells are going to take up the particles. So they actively take up, and we wanted to repeat this by doing a foot pad injection and looking at draining lymph nodes. And what you see is that at 48 hours, you have approximately 12% of cells in the draining lymph node have taken up the TMV particle. And we measured this for up to 15 days, and we believe that it could probably go as long as 30 days based on our data with the immune response profiles that we're getting. So we also wanted to look at what kinds of cells were taking up the TMV molecule in draining lymph nodes. So we had about a 50% expansion of the cellularity of the lymph node. So we have increasing numbers of cells, and we have both B cells increasing approximately by 50% and T cells. And this is a dose response, whether we use 10 or 50 micrograms of protein. We also looked at what kinds of markers were increasing in those draining lymph nodes, and we get some of the classical cell surface regulatory molecules. CD86 is an indicator of activation, increased class 1 and class 2. And in dendritic cells that are purified from draining lymph nodes, we also see an increase, a strong increase in class 2, which is an indicator of antigen crossover, as well as surface markers that indicate activation status. So our response peak in most of these measures was approximately 2 days, but we could still measure the persistence of activated cells far past the time when antigen had been taken up and carried away. So this is a cartoon, a ribbon diagram of coat protein. The inner molecule surface interacts with the plus strand genomic RNA, and conveniently both the N and C termini are exposed on the surface. What we originally did was we added genetic fusions to either the N and C terminus and got remarkable induction of antibodies and CTLs there. And this is a cartoon because what it shows is the density of array that we can provide by doing this kind of antigen display. So TMV has 21,007 coat proteins, and just based on the fact that we can see that all of the coat proteins are associated with a molecule, we can get a very high density load of any molecule that we can get to interact with the surface. So these are electron micrographs. This is a wild type TMV, and you can see the central RNA core. And when we conjugated protein to the surface of TMV, you can see that it's actually, you can see the protein lining up on the outside of the virus, and this is a cross-section where you can actually see the linker distance and the accumulation of protein around. So not only can we add peptides at very high density, but we can also associate protein. So the idea is that we're going to take weakly immunogenic subunit proteins that are defined antigens from pathogens and fuse them to TMV that has the capability of activating immune cell uptake as well as activate surface marker activation. So we know that TMV provides the size, repetitive antigen display, and even though the nucleic acid isn't replication competent, we believe that it provides a TLR signal because it is a plus strand RNA, and we also know that these molecules become endosomally located, which is where TLR7 is located also. So our first experiments were with influenza HA. The HA molecule is at a very high density on the surface of influenza, and it is the molecule that identifies the strain name. So H1N1 is one of the molecules that we were looking at. The amino acid changes are what dictate seasonal influenza shift. But what we're more concerned about in a public health matter is if we get a drift that goes into a shift and we get a completely different class of HA molecules that we haven't actually been exposed to. So we can actually measure neutralization of the virus by looking at antibodies against HA. So these are images of, I'll get to that in a minute. So we originally used an H1N1 HA molecule that Kentucky Bioprocessing produced at 500 gram scale. So they made a lot of HA protein. They were using the same plant expression system that is now being used to make the ZMAP antibodies. And what they found was that it wasn't very immunogenic by itself, which is no big surprise. They immunized. They had to put in 100 micrograms just to see a response. The other molecule that I've been working on is H5HA from the Vietnam bird flu strain. Their proteins are either monomeric or tetrameric, and it doesn't seem to matter what the source of the protein is. The H5 species was made in baculovirus, and I got very similar results. I'm using a chemical linkage that generates an amine, so no foreign chemical signature. It links the HA protein directly to the TMV surface. We analyze the conjugation products by SDS page and EM, and dynamic light scattering to tell us how big the particles are. And then we looked in mice for appropriate HA reactivity. So this is our typical analysis of a conjugation reaction. This is TMV coat protein, this is the HA molecule, and this is the H1 and H5 species. After a certain amount of time with the chemical conjugate, we lose the free HA protein completely. We generate high molecular weight species that are at least 2 million kilodaltons. And the majority of the material actually doesn't run into the gel. And we've tried MALDI to tell how big the particles are, but it's not, the size range is still too large. But you can see in both H1 and H5, this is a tetramer and this is a monomer. We achieve this high molecular weight and we lose our free HA. So we did electron microscopy to look at the diameter. The other thing we weren't sure about was whether or not the molecules were forming trains or mats or blobs or balls. And it really looks like the molecules are independent viruses that are coated with protein. And that was confirmed by dynamic light scattering, where we compared the average walking rate of TMV. It's an end over, it's a kind of a difficult model because it's 20 nanometers by 300 and they roll and they walk, so you don't actually get a round shape out of it, but we get an increase in the average diameter of the particle, and it also doesn't look like we have giant mats of protein. So the conjugation is, we think of it as coating the surface of the virus, so we're really making pseudoviral particles with a plant background, and they're not really cross-linked to each other, and they're just monodispersed viral particles coated with HA. So we wanted to look at vaccine potency. We wanted to look at boosting and the use of adjuvants that might enhance the immune response, virus neutralization, and protection in a challenge model. So our H1N1 vaccine generated, I can't actually read the scale over here. We were looking at microgram per ml of IgG. I know a lot of people use log scale, but We got extremely robust antibody responses with or without squalene adjuvant. And on this scale is the HAI titer. And you can tell that the animals that lived after one single dose of 15 micrograms of HA, the survivors all had HAI, but none of the guys that died did. So what we see here is that when we add squalene oil and water adjuvant to TMVHA, we can get 100% protection. And with no squalene, it's approximately 50%. And for most of the articles that I've read, no one gets any protection with one dose of HA under any circumstances, whether they use squalene or not. And this is the HA with squalene. And then for whatever reason, the H1 in this particular vaccine, the quantity was low after they measured it, but it offered almost no protection. When we did 2 doses, we got 100% protection of 2 doses of TMVHA, regardless of addition of squalene adjuvant. And we also saw much better induction of HAI titers. We also looked at whether or not antibodies that were induced could recognize other clades of H1, and we actually got pretty good recognition above 40, which is the cutoff. And we even got antibodies that recognized H5N1. So with H5, the titers were lower across the board for the single-dose vaccines, but the difference in survival was negligible. We still got about 50% survival with unadjuvanted TMVH5. We got 100% with squalene. And with 2 doses, we got 100% protection also. And obviously the HAI titers came up with the 2-dose vaccines. So what we've been working on recently is a plant-made H7HA, and it's a 2-dose study, and each of these groups is a 140-day interval. And what you see is that where you have an asterisk, you have significantly improved responses compared to the baculovirus partner or equivalent. So we wanted to compare it to what's actually being used as a vaccine. And for at least the first couple of months, we have much better augmented immune responses. And the TMVH7 is also much augmented compared to baculovirus without adjuvant. We also have strongly boosted IgG2A and B isotype and HAI using the TMV-HA conjugate vaccine. So we've recently been trying this with other model system antigens. We have achieved 100% protection with a 4-antigen combination of tularemia-specific antigens. And we've been doing intranasal delivery because that's very important for an intranasal pathogen. We also had 100% protection against Yersinia pestis, which is a plague pathogen. Also, intranasal delivery, and in this case, we were able to get protection with no adjuvant. And lately, we've also been working on a multi-TB antigen vaccine, and we have been getting extremely high CTL responses, better than antigen plus adjuvant, and we're just in the process of starting challenge studies for the TB vaccine. So we also are using TMV to carry nucleic acid into cells, and that's something that I'll talk about maybe next year. So we met our goals for the study. We got HAI, we got protection, and we're confident that we can work backwards from protection to full protection in single-dose vaccines because we haven't done any dose modulation. We haven't really played around at all with adjuvants, and we're hoping to also shift completely to intranasal dosing. So thanks to my colleagues who participated in this study, and I'll take questions if you have any.